Hello and welcome. In this video, we are going to discuss bias in clinical research. I'm not a biostatistician or epidemiologist, but this video is based on my personal study about research and my experience in research over the past 40 years. At the end, I will give you some special tips on this subject. So let us start. We will cover the following topics in this video. First, we will talk about what is the meaning of bias and what is the meaning of bias in clinical research, and talk about the different types of bias in clinical research and how we can remove bias from our research protocols and research methodology. In simple terms, bias means having a preference or a leaning towards something or some person. This can happen when we like one thing or group more than another, and it can affect how we make choices or share information. Bias can show up in different ways, like when we favor our own ideas or treat one group better than another without a good reason. It can make things unfair and lead to misunderstandings. It's important to be aware of bias and try to be fair and objective in areas especially like news, science, and also in everyday life. In clinical research, bias refers to a situation where the study or the results are influenced in the way that is not fair or impartial. This can happen when certain factors or preferences affect the research and cause it to be less accurate or reliable. Bias can make the research results misleading or not truly represent the real situation. To do good clinical research, it's important to try to avoid bias and be as objective and fair as possible in the study and its findings. Here is a detailed overview of different types of bias in clinical research. First, we come to selection bias. This occurs when the study sample is not representative of the target population. This can result from a non-random selection of participants or the exclusion of certain groups. An example is a clinical trial recruiting only healthy individuals for a drug study may not accurately represent how the drug performs in the general population, which can lead to a biased result. Another example is information bias. Information bias, also known as measurement bias, happens when there are errors in the measurement of variables or outcomes. It can be categorized into two subtypes. A is misclassification bias, or inaccurate categorization or measurement of exposure or outcomes. An example is, in a cancer study, if the method of diagnosis is not standardized or is subject to interpretation, it can lead to misclassification of the disease status. Another subtype is recall bias. It occurs when participants do not accurately recall past events or exposures, often leading to an overestimation or underestimation of risks. A very common example of recall bias is doing verbal autopsy for maternal mortality. So an example of number 2 is that in a case-controlled study investigating the link between smoking and lung cancer, patients with lung cancer may be more likely to recall smoking habits due to their illness, leading to an overestimation of the association. So this is another example of recall bias. Another kind of bias is observer bias. This takes place when the researcher's expectations, beliefs, or prior knowledge influence their observations and data collection. For example, in a clinical trial, if the investigators are aware of the way that the treatment is assigned to their sample population, they may unconsciously record outcomes in a way that favors the treatment which they own personally believe to be more effective. Publication bias can occur when the research findings that are statistically significant or positive are more likely to be published, while negative or non-significant results remain unpublished. An example is a clinical trial with a promising result may be more likely to be published, leading to an overrepresentation of positive findings in the literature. Confounding bias is a very important bias that happens when we are planning studies. This means that an extraneous variable, which means a variable that has not been taken into account, has not been considered in the study. And this will affect the relationship between the exposure and the outcome. It can lead to incorrect conclusions about causality. An example is in a study examining the relationship between coffee consumption and heart disease, age could be a confounding factor, since older people are more likely to have heart disease and also may consume more coffee. Lead time bias. This occurs when early detection methods, not improved outcomes, create the improvements of improved survival. In other words, if supposing you have some kind of test or some kind of methodology by which you are detecting the disease early, then this may lead to a better outcome. And in such cases, the treatment of these cases may not be the reason for a better outcome, but it's just that the disease was detected early. An example is in a cancer screening study, earlier detection of cases that may seem that the screened group has better survival rates, while in fact, it is just the lead time between diagnosis and death that has changed. Length time bias happens when slow-growing or less aggressive cases are more likely to be detected by screening, leading to a higher apparent survival rate. For example, a mammography program may detect more cases of indolent breast cancer, making it appear as if the screening is more effective than it is. So the next question is that how can we avoid bias in clinical studies? Avoiding bias in clinical studies is paramount to ensure the validity and the reliability of research findings. Bias can compromise the integrity of the study and lead to incorrect conclusions, potentially impacting patient care and treatment decisions. Randomization is like a guardian angel. It is one of the most potent defenses against bias. When patients are randomly assigned to treatment groups, it helps ensure that each group is comparable and reducing the risk of selection bias. This randomness plays the level field and creates a foundation of impartiality upon which the trial can stand. Blinding is another crucial measure, akin to wearing a blindfold while tasting food to prevent bias from one's preconceived notions. In a double-blind study, neither the participants nor the investigators know who is receiving the treatment or the placebo. This secrecy prevents observer bias and patient expectations from affecting the results. Placebos are the great impersonators, and they are used to test the efficacy of a new treatment. They look and feel like the real thing, but they are inactive. In this way, participants in the control group are not aware that they are not receiving the active treatment, reducing expectancy bias. Placebos help isolate the true effects of the treatment being studied. For publication bias, researchers can combat the tendency to publish only positive results by pre-registering their study protocols and analysis plans. This commits them to publish the results regardless of the outcome. Additionally, journal editors and reviewers play a critical role in promoting the publication of well-conducted trials, regardless of their findings. Sponsorship bias is a very painful subject, and funding transparency is the key to avoid sponsorship bias. Sponsorship bias means that if a drug company sponsors a research project in which they are using their own drug, then there is a possibility that everything and the results will go in favor of the funder group. Disclosure of funding sources and potential conflicts of interest is a cornerstone of ethical research. Independent, non-industry funded research can also be promoted to minimize the influence of sponsors on study design, analysis, and reporting. Another way to avoid bias is to study systematic reviews and meta-analysis, which act as detectives researching for bias in the vast landscape of clinical research. By aggregating and analyzing the findings from multiple studies, they can identify inconsistencies and potential biases, and they help to arrive at a more accurate conclusions. Systematic reviews and meta-analysis have another important function as they highlight areas in that topic on which clinical research should be conducted and performed. So they identify gaps in the knowledge. So whenever you do any kind of research, whether it is community-based research or whether it is hospital-based research, it is important that your research and your trial is being monitored by your local data and safety monitoring board. These serve as impartial overseers. These independent experts review the accumulating trial data and ensure that the trial remains ethical and safe. Their vigilance helps protect against sponsor bias and assures the well-being of the participants. So in summary, clinical research is a noble pursuit seeking answers to improve human health and well-being. The fight against bias is an ongoing battle, and researchers arm themselves with the tools of randomization, blinding, placebo controls, independent oversight, transparency, and the scrutiny of systematic reviews to ensure that their findings are untainted by bias as humanly possible. In this way, they honor the sanctity of science and the trust of the patients who make these investigations possible. So in the end, I will just give you a tip that the most important thing that can cause bias is lack of randomization and the lack of ensuring that all relevant variables have been identified when you're planning the research. So if you plan the research and you randomize your subjects, and this is only if you're doing an interventional research, and if you blind your study, then the chances are that you can get a very good and authentic information, which is likely to be published in good journals. If you like this video, please subscribe, press the like button, comment, share, and press the bell icon for notification of future videos. Thank you and goodbye.